Transformation is one of the three processes through which bacteria can obtain DNA from the environment. This strategy has been deeply studied and several protocols have been developed so as to take advantage of transformation to introduce plasmids and other types of genetic material to several types of organisms. In this way, the expression of proteins of interest can be induced and controlled. In this practice, we encompass two types of transformation strategies: electroporation and chemical transformation. We obtained efficiency values that fall below the commercial standard, but the cell count was affected by the presence of uncountable colonies.
Transformation is the uptake of DNA from the environment by a bacterial cell. Scientists adapted transformation for propagation of plasmid DNA, protein production, and other applications. In research, transformation introduces recombinant plasmid DNA into competent bacterial cells that take up extracellular DNA from the environment. Some bacterial species are naturally competent under certain environmental conditions, but competence is artificially induced in a laboratory setting. On the other hand, transfection refers to the process of artificially introducing DNA or RNA into mammalian cells by means other than viral methods. Most of the times, the piece of DNA of interest is ligated into a vector that can be plasmid, cosmid, bacterial artificial chromosomes, yeast artificial chromosomes, or even human artificial chromosomes. The most common transfection methods are chemical-mediated transfections such as calcium phosphate co-precipitation, liposomes, and physical methods such as electroporation, gene gun technique, and microinjection. Basically, the main difference between transfection and transformation is that detransfection refers to the introduction of foreign DNA into mammalian cells, while transformation refers to the introduction of foreign DNA into bacterial, yeast, or plant cells. When choosing a transformation method, it is important to take into account several aspects in order to get consistent, useful results. For example, the vigorosity of the cells. It is necessary that the culture to be used is in an exponential phase, and that it has been sowed just a few hours before starting. You can use the morbidity measurement at 600 nanometers to have a reference of the appropriate optical density. The sample quality. DNA should not contain phenols, ethanol, or detergents that damage the cell membrane and inhibit microbial growth. Neither should it contain salts, since these conduct electricity, causing the death of the cells in electroporation. Amount of DNA. The amount of DNA recommended is from 1 to 10 nanograms up to 5 microliters from 100 microliters of cells. In the case of electroporation, less DNA is used than in transformation by chemical method. The next one is the efficiency. Depending on the purpose of the transformation, it is important to choose the best method to achieve the desired efficiency. For example, in the case of electroporation, efficiency rates are higher than the ones obtained by thermal shock, which is important if it is desired to transform ligations. And the next one is the recovery time. Regardless of which method of transformation is used, outgrowth of bacteria following transformation allows repair of the bacterial surface and selection of recombinant cells if the newly acquired DNA conveys antibiotic resistance to the transformed cells. There are more than one method to make the transformation of a bacterial cell. Here we have some examples of bacterial transformation methods. Biolistic technology. The biolistic technology, which is also called particle bombardment, is a direct physical method of introducing nucleic acids into cells. Nucleic acids and other biological molecules are coated into high-density gold or tungsten nanoparticles, microcarriers, which are then accelerated to high velocity by a helium pulse and driven in through the cell walls and membranes into the target. The physical nature of this technology makes it extremely versatile and easy to use. It can be applied to a wide range of targets, including cell cultures, tissue, organs, plants, animals, and bacteria, as well as organelles. Another example is the plasmid acquisition mechanism based on the Yoshida effect. When a colloidal solution containing nano-sized acicular material and bacterial cells is stimulated by sliding friction at the interface between hydrogel and an interface-forming material, the frictional coefficient increases rapidly, and the nano-sized acicular material and bacterial cells form a chestnut burr-shaped complex. This complex increases in size and penetrates the bacterial cells, thereby forming a penetration intermediate, due to the driving force derived from the sliding friction. And the last example could be the electrospray technique. This process was previously known as electrohydrodynamic atomization. Electrospray employs an electric field to disperse and accelerate liquid droplets or fine particles. An advantage of this method is that it doesn't require prior treatment for competent cells. The principle employs gold nanoparticles to be conjugated with DNA that can increase the momentum of the plasmid during electrospray, resulting in higher amounts of penetration through cell wall.
Before carrying out the following protocol, it is necessary to sow a strain of Escherichia coli in LB plate and incubate overnight at 37 degrees Celsius. On the second day, a colony isolated from the top 10 strains is seeded in 5 ml soft medium and incubate at 37 degrees Celsius with shaking overnight. On the day of the practice, at least 3 hours before starting, 100 ml of LB medium will be sown with 1 ml of the seed culture and incubate at 37 degrees Celsius. Preparation of component cells. 1. Take 0.3 ml of seed culture of top 10 bacteria, so 30 ml of LB medium. 2. Incubate at 37 degrees Celsius with shaking for about 3 hours until the optical density reaches an absorbance of 0.5 to 0.6 at 600 nanometers. 3. Place the flask with the bacteria culture on the ice for 10 minutes. Finally, divide the culture in half to obtain competent cells using 2 protocols. Centrifuge each part of the culture until the bacteria tablet is concentrated. 6,000 revolutions per minute, 4 degrees Celsius, 5 minutes. Once the cell pellet is obtained, remove the supernatant. Resuspend in 10 ml of cold 0.1 molar calcium chloride and incubate on ice for 10 minutes. Centrifuge at 6,000 revolutions per minute, 4 degrees Celsius, 10 minutes. 3. Remove the supernatant and resuspend the bacterial tablet in 2 ml of calcium chloride 0.1 molar to 20% glycerol solution. Incubate for 10 minutes on ice. And finally, prepare a liquid of 200 microliter and store at minus 70 degrees Celsius. 1. Once the cell pellet is obtained, remove the supernatant. Resuspend the pellet of bacteria in 10 ml of the cold 10% glycerol solution. 2. Incubate on ice for 10 minutes and centrifuge at 6,000 revolutions per minute, 4 degrees Celsius, 10 minutes. 3. Remove the supernatant and resuspend the tablet in 2 ml of cold 20% glycerol solution. And finally, prepare a liquid of 200 microliter and store at minus 70 degrees Celsius. 1. Take a liquid of frozen chemiocompetent cells and place it on ice immediately until it thaws. 2. Add 50 nanogram of plasmid of no concentration. 3. Incubate on ice for 10 minutes. 4. Place in the thermoblock at 42 degrees Celsius for 45 seconds. 5. Place on ice 5 minutes. 6. Add 300 microliter of soft medium under sterilized condition and incubate at 37 degrees Celsius for 30 minutes. with starting at 250 revolutions per minute. 7. So, two different dilution volumes from the previous culture in LV plates with ampicillin and incubate for 16 to 20 hours at 37 degrees Celsius. And finally, the next day, count the colonies that grew and calculate the efficiency of transformation. 1. Taking a liquid of frozen electrocompetent cells, place it on ice immediately and let it thaw on the ice. 2. Placing a 1.5 milliliter tube, a mixture with a total volume of 40 microliter that includes the electrocompetent bacteria and 50 nanogram of a plasmid of no concentration. 3. Transfer the 40 microliter of the above mixture to the 0.2 centimeter electroporator cell and plate it on ice while programming the electroporator. 4. Place the cell in the electroporator chamber and give the electrical pulse. 5. Transfer the mixture to the 1.5 milliliter tube and add 300 microliter of soak medium under sterilized condition and incubate at 37 degrees Celsius for 30 minutes with shaking at 250 revolutions per minute. 6. So, two different volumes at the previous culture in LV plates with ampicillin and incubate for 16 to 20 hours at 37 degrees Celsius. And finally, the next day, count the colonies that grew and calculate the efficiency transformation.
In Team 5, transformation by electroporation, observed in Figure 1, shows a large number of colonies, while the chemical transformation in Figure 2 shows a smaller number of colonies. In both cases, the colonies are reported as uncountable, and therefore the transformation efficiency calculation is not performed. In Team 6, a greater number of colonies is observed in the chemical transformation. Nevertheless, when counting colony-forming units, a total of 147 was obtained in the chemical transformation, and a total of 366 was obtained in transformation by electroporation. Then the transformation efficiency is calculated. For the calculation of transformation efficiency, the following formula is used: colony-forming units over nanograms of DNA times conversion factor times dilution factor. In the practice, a total of 100 nanograms of DNA was used. In chemical transformation, 40 microliters of cells plus 2 microliters of plasmid and 1,400 microliters of soak medium were added. Then 200 microliters of this cellular medium were taken. This consists in our first dilution factor. After this, the soak medium was added until reaching 1 milliliter. From this quantity, 100 microliters were put on agar. It is our second dilution factor. The chemical transformation efficiency was the following. The same efficiency calculation is repeated with the transformation by electroporation. The only value that is different, besides the colony-forming units, are the 38 microliters of cells used. Transformation by electroporation efficiency was the following.
Transformation is a technique that allows foreign DNA to be introduced into a cell, usually a bacteria. This methodology is very important for microbiology and genetic engineering because it can be used for DNA sequences or plasmids storage and replication. Plasmids usually contain a bacteria replication ORI and an antibiotics resistant used for a selection marker. In order to improve the transformation efficiency, competent cells must be used. Electroporation and chemical transformation are two of the most used transformation methods. The first one takes advantage of the cell's membrane lipids negatively charged, same as the DNA phosphate groups, both repelling themselves. Therefore, salts such as calcium, rubidium, potassium, among others, are used to neutralize the negative charge, and after so, a thermal shock is induced to create channels on the membrane that will allow DNA to go through. For electroporation, an electric pulse is applied on a short time, causing instability of the cell's membrane conductivity and permeability, allowing DNA to be introduced. For this procedure, host cells and selected molecules are suspended on a conductive solution, creating an electric circuit where an electric pulse is applied only by few microseconds. These two techniques have different methodologies. For electrocompetent cells, they need to be resuspended on a 10% glycerol solution, while chemiocompetent cells on a 0.1 molar calcium chloride solution. Electroporation is known to have a better transformation efficiency when using cDNA, ligations, or genomic DNA. It also allows to use small amounts of sample. It is required to have a pure and salt-free DNA while performing an electroporation to assure that there is no extra conductivity that could kill the cells while experiencing the electric pulse. For chemical transformation, pure samples of DNA must be used. Pollutants such as phenol, ethanol, isopropanol, detergents, and proteins reduce the transformation efficiency.
In the laboratory, top 10 strains of E. coli were used, which according to Thermo Fisher Manual of Invitrogen have an efficiency plus than 1 per 109 colon-formers unit per microgram. In one shot, top 10 E. coli chemical competent. In a multi-shot, top 10 E. coli chemical competent coli and efficiency plus than 1 per 108 colony-former unit per microgram is observed, while in electrocompetent top 10 E. coli in the convenient one-shot format, an efficiency of more than 1 per 109 colony-former unit per microgram is reported. According to our results, the reported efficiency is not met. There was less efficiency in our chemocompetent cells, 1.059 per times 10 to the 5 potens, and a higher efficiency in our electrocompetent cells of 2.63 times 10 to the 5 potency colony-formers unit per microgram. As mentioned above, in electrocompetent cells, there is higher transformation efficiency. Strains of E. coli are regionally electrotransformed to achieve efficiency of transformants 10 to the 9 potency and 10 to the 10 potency transformants per microgram of DNA. The transformation frequency can be as high as 80% of the surviving cells. There are steps that could have failure that therefore not achieve the expected efficiency. In the chemical transformation, it is observed that there are many colonies that are not isolated and therefore cannot be counted. However, at first sight, it is noticeable that in this plate there is a higher transformation efficiency. This was due to that in the preparation of competent cells at the time of adding soak medium, our microtube slipped and much of the content was lost. Another mistake occurred was the lack of resuspension of the cell pallet at the time of adding either CaCl2 at 0.1 molar, 20% glycerol, or 20% glycerol gold. It is important to have more caution and better pipetting control.
The principal difference between the two ENCOMPASS protocols relies on the form in which the cellular membrane permeability is modified. On the chemical transformation, the negative charges of both the membrane phospholipids and the DNA are neutralized through the addition of a salt, favoring the entrance of genetic material once there is no charge limitation. This happens through a heat shock that forms temporal hydrophilic pores. In the case of electroporation, salt is removed from the media, giving place to an environment of low conductivity but high resistance. In this case, an electrical current of high voltage is conducted through the media for a very short time, not only favoring the entrance of DNA to the cell, but also ensuring that bacteria are kept alive. At the end of the practice, the main limitation was to calculate the efficiency due to the presence of uncountable cell colonies. A solution to this problem would be to repeat the inoculation process with larger dilutions.